We're very happy to introduce Dr. Maria Ruiz Ortega. The title of her talk is Learning Predictive Signatures of HLA Type from T-Cell Repertoires. And just a little bit about her. She's a physicist interested in mathematics behind natural processes. She studied at the University of Valencia in Spain, and after completing her master's in theoretical physics, she realized she was more passionate about cells than particles. So consequently, she moved to Paris for her PhD, where she was supervised by Alexandra Walczak and Terry Mora at École Normale Supérieure. Her research focused on the quantitative analysis of various aspects of the immune response, including the diversity of lymphocyte receptors and their changes after pathogen exposure, with the ultimate goal of detecting antigen-responding cells. And currently, she's in her first year of postdoctoral research with Marta Luska at Mount Sinai Hospital in New York City, developing theoretical models to predict viral evolution that could improve the design of new vaccines. Thank you so much, Maria, for joining us. We're really interested in what you have to share today.
Yeah, sure. Thank you so much, Monica, for the presentation. Yeah, hello everyone. I'm Maria. I'm gonna tell you about this little collaboration I did last year with the team of Paul Thomas at Memphis St. Jude Hospital. And it's focused on this HLA prediction from repertoires. So yes, oops, okay. Just a little introduction. I mean, I guess everyone is familiar with it, but we are gonna focus this talk on the adaptive immune system. The immune system is an ensemble of resources that our body has to protect against external pathogens. There are two types of subsystems in the immune system. There is a more primitive and general and non-specific response, which is the primary immune response. But then there is another one, way more sophisticated, that can evolve in time. I can really detect the kind of pathogen that is entering our body and design a specific response, which is the adaptive immune system. This is the subsystem we are gonna focus in. And two of the main actors are the two kind of lymphocytes, which are B and T lymphocytes. We are gonna talk about the T cell response in this case. So imagine you have an organism and it's attacked by an external pathogen. The first thing that is gonna happen is that one cell called the dendritic cell of this more general immune response is gonna eat the cells that have been attacked by this pathogen. And then this dendritic cell is gonna present in his surface some small protein fragments from this pathogen that is called the antigenic peptides. Then there will be some cells that I call these T lymphocytes that will detect that these antigenic peptides are not part of our body, and this will trigger an immune response. So these T cells will get activated, they will proliferate, and they will attack other cells that might be infected, and also will protect the body against it. So as I said, when the T cell recognizes this antigenic peptide, we have the immune response. But for this recognition to happen, we need two conditions to be fulfilled at the same time. So as I said, this is like the dendritic cell is presenting this antigenic peptide. The presentation of the antigenic peptide only happens in this protein structure called MHC complex. So for the T cell to get activated, it needs to recognize first this MHC complex. To do so, the receptors of the T cells have some kind of specificity that is given by a gene called HLA gene. We have two types of HLA genes. We have the class 1, which is A, B, or C, and the class 2, which can be all these combinations. And inside each type, we have all these possible alleles. We have some alleles coming from the mom, some coming from the dad, and considering all the possible combinations, we would say that the HLA specificity of a person is quite unique. To ensure that the receptor is specific to this MHC complex, once the receptor has been created in the thymus, is gonna be exposed to different antigen-presenting cells that could be dendritic cells or B cells. So only those T cells that can recognize the MHC complex will be retained by the thymus, and the other will die by apoptosis. Also, depending on the type of MHC complex that they detect, they will specialize in two different types of T cells. This we will speak later. So then we have once then in this time selection we ensure this MHC specificity, but we also need the T cell to be able to recognize the peptide. To do so, our body has a super sophisticated mechanism called BDJ recombination to create the T cell receptor. This BDJ recombination is an stochastic process where randomly genes B, D, and J are selected from the entire pool of possible genes, and then they are merged together. But at the same time of this merging, there is some also random insertions and deletions of nucleotide pairs that are gonna be like inserted among these V and J junction and D and J junctions, and then we are gonna end up with a nucleotide sequence that is highly variable. There is estimations that this process can create up to 10 to the 70 different possible receptors. So in this way, we are ensuring that the receptor is going to be specific to almost any kind of pathogen that is going to enter. So we have this peptide specificity also ensured thanks to this BDJ recombination. So in this, like to summarize, when there is a immune response, there is some kind of love triangle between the peptide and the T cell, where the T cell need to recognize at the same time to this peptide, to the MHC. Also, the peptide needs to be specific to the MHC. So this triangle is called the TCR peptide MHC restriction. There is like a lot of efforts right now to try to determine out of the receptor sequence of the T cell what is the peptide specificity. And there are not that many works knowing what is the MHC restriction of the receptor, and this is what this work is focused on, to disentangle given a receptor what could be the MHC specificity of it. We are going to try to do that through some statistical analysis. To do so, well, but first of all, we need to know why the basis of this statistical analysis will be to try to find TCRs that co-occur among different people. So this, as I told you, there is like this VBJ recombination process has the availability of create almost an infinite amount of possible receptors. So just by statistics, one might think that a receptor is completely unique and is not going to be repeated among people, but this is not true. There is if we take the list of receptors of different individuals, we see a systematic sharing of different receptors. Why is this sharing happening? Well, first of all, because this BDJ recombination process is not completely equally likely. So some receptors have the probability of creating them is much higher than the probability of creating another, because we have different scenarios to create these receptors, and some scenarios are very likely to happen and others are not. Then Also, as I said, in the thymos, we have some selection. So across all the possible receptors that we are going to have, the thymos is going to select a subset of them. So this is going to decrease also a lot the heterogeneity of the possible receptors. And now we have a third and fourth conditions that are deeply related among them, which is, first of all, the antigen-driven selection. So when a person is attacked by, different people are attacked by a common pathogen, they will trigger a similar response because they will have like similar TCRs responding to the same antigen. But as I said, there is some restriction. There is this MHC restriction. So if we see the same TCR happening across different individuals, it can only happen if these individuals have the same MHC, because only in that way they will present the same peptides and they will have the same TCR recognizing. So we are going to focus on people who have suffered some common antigenic exposure, and they have the same TCR's response. This could mean that they have the same MHC happening. I don't know if this is kind of clear. So to do so,
That's clear. That works.
okay, so here is where we start with our sequencing data sets. We have a list of 1,039 individuals for which we have the entire sequencing repertoire. So we have a list of the TCRs. Not all of them, like for like around half of them, we have both the alpha and the beta chain. For the other half, we only have the beta chain. And we also know the MHC alleles, that they are positive on each individual. So once we know these two things, we are going to create what is called a contingency table. So this table is just something like that. It's a statistical resource. So for each for each possible combination of a given TCR receptor in a given HLA, we are going to compute how many people are positive for both, how many people are negative for both, and then how many people are positive only for one. So they have the TCR but not the HLA type, or on the other way around. And once we have this contingency table, we are going to perform a so-called Fisher test, which is telling us how likely is that these combinations, I mean, these configuration is happening just by chance, or is happening because these HLA and TCR are related among them. So this Fisher test is going to give us a p-value. If the p-value is close to 1, it means that probably this is the two conditions are not related among them. But if the p-value is very small, it means that probably the occurrence of one thing, for example, the occurrence of having this TCR, is gonna bias the occurrence of also having the HLA. So what we are gonna do is compute all the possible tables or the possible combinations of TCR and HLA and perform this Fisher test and filter out but the ones that have a p-value smaller than 0.05, which means that these two combinations are possible related and that the TCR is specific for a given HLA type. So once we have performed these Fisher tests for all the possible combinations, as I said, we filter them and we get a resulting list of thousands of TCRs that seem to be related with a given HLA type. A sanity check of this, for example, as I said before, the TCRs, depending on if they relate to MHC complex type 1 or type 2, they are called CD4 or CD8. This is the specificity of the co-receptor. So as a sanity check for this list that we obtained, for some of them, we knew if the TCR was belonged to a CD4 or a CD8. So what we did is to check if this Fisher test was giving us specificities of HLA type 1 for the CD8 and HLA type 2 for the CD4. And we saw that in general, although there was some small residues, the test was pretty accurate, and we were obtaining that HLA type 2 was related to CD4 and HLA type 1 was to CD8. So it seems that the Fisher test was working properly. Then another thing that we do is like with this list of TCR that are specific to this HLA, we learn a simple logistic regression model. So what we do is to learn some weights, assign some weights on the presence of or absence of the sequence to try to determine if the person is positive or negative for a given HLA. So from this list that we have of TCR to be specific to HLA, we divide it between 70% of training and 30% of testing. And in this 70%, we use our algorithm to learn the weights. And the algorithm is going to assign, is the beta, is a weight to each of the sequence. And then in the testing, it's gonna give a 1 or a 0, depending on if the how many TCRs specific are detected in this repertoire, then we can say if the person probably is positive or negative with some HLA. Then we try this logistic regression model in an independent data set that is for which we also knew the HLA type, and we try to predict them. So this third data set was a cross-validation data set that we didn't learn anything from them, and still we could predict some HLA types, as you see here, with a lot of accuracy. The bad news is that we could only do it for like very common HLAs. It's very difficult when the HLA doesn't appear very frequently, because then there is no statistical data to do these assignments. Another thing that we did is like we separate the TCR that were found to be related to a given HLA. We separate them by HLA type. So for example, here is for HLA type 1. We took all the TCRs related with HLA type A, the HLAs type B, and the HLA type C. And then we did some cluster analysis. So we took the CDR3 sequence, which is the most variable part of the TCR, and we clustered them in some networks of coming distance 1, which means that two dots that are related are CDR3s that are only one amino acid away. So we see that TCRs that are very similar among them tend to respond also to the same MHC type. So if we have here, you see very clean cluster, for example, all these responding to A0201 are TCRs that are very similar also at the level of a structure. So we could see some that this specificity is already determined at the level of the structure of the PCR sequence. So considering that similar TCRs tend to be specific to the same HLA type, we also did this. We cross-validated our clusters to see if the same TCRs, these TCRs that are specific for some HLA, they were also specific for some peptides. And it's true that these kind of analyses are very dependent on how many peptide database you have, but we could find some of them. And for example, all these clean clusters of TCR that respond to the same HLA, they also respond to the same peptide. These were for flu, and some of them were also for Crohn's disease. And yeah, I think these are the ones that we used. I didn't add it, okay. But what we did also, as I said, considering that similar sequences respond to the same HLA, we did some variation of this HLA detector of this program, and something like this. So what we were doing here is to learn to assign some weights to sequences that we found to be specific. So the variation that we included is like, instead of taking only the sequence as it is, we took also all the possible variations of the sequence. So we took a given sequence, for example, this one that is be related with this HLA, and then we introduced all the point mutations possible. And we consider that all the sequences that are one mutation away will be also specific for the given HLA. And we rerun this predictor, and we saw that introducing this 1-point mutation, the prediction of the HLA was even better than considering only one kind of sequence. So this, I didn't add it. And then there was like, I think the most useful application of this analysis that was to try to to apply this predictor to untyped data sets. So we started some iterative process of typing untyped data sets and learning some information from these untyped data sets. I will ask a bit more slowly how it goes. So we started with our list of sequences that we obtained, this list of sequences that we obtained from our data set from which we know the HLA type, and we took a new data set from which we don't know any HLA type for the patients. And we split this new data set in, it was like around also 1,300 individuals in, no, 1,500, sorry, individuals. And we split it in 15 groups of 100 individuals. So for the first 100 individuals, we used our original logistic regression model to type all these 100 individuals to see if they were positive or negative for a given HLA. We tried with A0, A0201, that is the most common, but also for DPV10401 and for two more, that is like C0702 and some HLA type 2 that I don't remember. So we use the logistic regression to type these 100 patients. From this logistic regression model, we obtain a list of individuals that will be positive and a list that will be negative. From the list of positive individuals, we rerun this Fisher test that we did at the beginning, and we obtain, considering that they were positive, we obtain a new amplified list of TCRs that are related to the HLA type. And we use this new list again to type the second ensemble of 100 patients. And we do this iteratively, so in which in each iteration we have more patients that are tested positive, and we will have a list of TCR related that is getting bigger and bigger. So one might be a bit skeptical about this process, because if the predictions are not well done, then all the TCRs that we finding to be related will be grown and will be accumulating on time. So to check if these new TCRs that were getting learned from untyped datasets were good, we took the list of the TCRs, these resulting TCRs from the Fisher test that we did on untyped datasets, and we apply the classifier. We train the classifier with two lists, with the list of original TCR sequences learned from typed patients, and with the TCRs learned from untyped patients. And we applied from these third data sets for which we didn't know anything. And we saw that with both, with the TCR length from typed and untyped, were very good, and with the combination of both of them, the accuracy was maximized. So it seems that the information we are learning out of these iterations is good quality information, and it's giving us, I mean, in each iteration you can see like the number of TCRs is growing exponentially, so we can end up with a huge amount of TCRs to be specific to a given HLA. And yeah, this is a bit complicated. Just ask me any question if you need a bit this iterative process. If it's not clear, I can try to explain it better. But This tool is called HLA Guesser. It's on GitHub. I forgot to add the link, I think. Yeah. But it's a very easy tool. You can upload your repertoire with alpha and beta sequences or with just one of them. And the predictor will give you a list of HLAs that will be positive or negative and some parameters to see the accuracy of the measurements. And it's very easy to use, and I would be very glad if people start using and I can add more data, because this could, as you have seen, the more data you have, the more precise it gets and the more information we get about these specific TCRs.
Maria, when
So
did you guys launch that HLA guesser on GitHub?
that was end of January.
Great.
Yeah.
And how many participants or how many people have been using it? Has it been a lot, a little? There been a lot of interest?
I mean, yes, now I guess it's normally it's been like 6 months already, but at the beginning I received a lot of emails with, because some, the typical installation that some people maybe cannot install it and so on. So almost every 2 weeks or something, I was receiving a couple of emails of people asking questions. Also, people who seem to have their own blood sequenced. They were trying to test it because they knew their HLA type and they have their sequences, and they were using their own blood to get it. But yeah, this is a bit of a work in progress, because there are not so many data, not so many repertoire data with HLA type. So
Yeah.
the more I can introduce the more powerful it's gonna get. But yeah, I would like to keep on working on that to make it more and more precise. But it seems that people are using it. Yeah.
Great.
And yeah,
Yeah, it sounds like a very interesting tool.
this is all, as you have seen, that it's as with a simple statistical analysis, you can predict some specificity, HLA specificity for a T cell receptor. This is I think it's an important tool, because many autoimmune disease or other kind of disease seem to be related with the HLA type, and there are a lot of, as I said, there are a lot of sequencing data out there for which we don't know the HLA type, and it would be nice to have a tool to get this out of data that were sequenced long ago, that we are able to determine the HLA type. And also, yeah, I mean, knowing the HLA restriction of a person, it's something needed to develop any personalized medicine. So I
All right.
think,
Hey,
yeah.
ask a quick question. This is
Yeah.
Todd.
Yeah, sure.
Thanks, Todd.
I'm curious about whether your analysis, when you have situations where you have heterozygosity at class 1 or class 2, do you have any idea whether that one of those genotypes is primarily shaping the repertoire?
I didn't get it. Sorry, could you repeat it?
Yeah, so I guess the question is, if someone is heterozygous, say they carry, let's just use type 1 HLAs as an
Yes.
example, DR3 and 4, do
Yes.
you have an idea whether that's dominantly shaping the CD4 repertoire towards one or the other? Do you
Mm,
see that in your
Uh,
data?
It's a great question.
y- Yeah, that's a great question. Actually, that's interesting. I don't know. My guess is that, yeah, probably it's okay. I think it's also not included here, but we did some analysis of comparing repertoires on people having different HLA types. And for example, we could see some signal of depending on the HLA type, there was some V gene usage that were like was predominant versus a V gene that was used for another HLA. So it seems that also the HLA type definitely is shaping how the repertoire is gonna look at a general level. Yeah.
Oh, thanks. This is a really useful tool. Appreciate it. Thanks.
Mhm.
Yeah, it's a great tool. And actually, Todd Brusco and Aaron Michaels together put this, are sort of spearheading this TCR, T1D focused TCR data repo. And so,
Mm-hmm.
yeah, so that is growing, and it might be a really great, this tool might be very interesting to use in concert or to kind of query that repo as it grows. I
Yeah.
wanted to see, I see Luke Tayton is on the call. Luke, do you have any questions? Oh,
I
here you
always have questions, you know.
Good.
Again, I think the primary data, the data sets are just too small to do anything with high confidence for now, but it's certainly worthy of pursuing. I think that, 2-3 years from now, those type of approaches on analysis that are reasonably simple, will be very fruitful. So we have to keep accumulating primary data.
Yeah, definitely, definitely. So, for example, yeah, this is a bit of one of the questions that I got from some user that tried to use it on himself. He got detected some HLAs that were pretty rare, and he was very surprised. But other HLAs were not detected, and it's always difficult because in the end, there are out of these 3 data sets, I get a lot of TCRs, but not that many. So finding these specific TCRs and then finding them in someone and being able to predict so far with the amount of data we have is small, but I think, as you said, that is growing fast. So the available data sets grow, this is gonna, it just take a couple of iterations to get very, very good results.
Yeah,
So
the one piece that is kind of missing on it's on us. Less and less people are trying to get TCR peptide MHC structures, because it's still very, very difficult to get those on. If we had structural information to back up your prediction, that would be
yeah,
far better. But again, we have about 400,
yeah,
500 structures.
yes.
Many of them are related to each other on that information that is very precious. Nobody is ready to go get it because
Yes,
it's
yeah.
too
For
difficult.
example, one thing that excited us was like, for example, some collaborator of us, he developed some tool to try to predict out of just the structure of the TCR, the CD4 or CD8 classification, and the results were pretty underwhelming. So he concluded that out of the extractor, there was no way of getting the CD4, CD8. But then here you kind of have it. I mean, you can see, it's a bit of a collateral effect, but first out of the sequence, you can see the HLA specificity and then you can get it. So I think this is a small signal. So there must be something out there in the structure that, so.
Yeah, but there again, we have no structure. We have no good structural information of
No, not at all. Yeah, I think it's
peptide MHC TCR on the co-receptor. So those,
yes,
yeah,
yes,
yeah,
yeah. It's fortunately it's a field that it's evolving super fast.
yeah.
But so far it's still the big gem of
In any
one of the
case,
big
keep it
gems
up.
of.
Keep it up.
Yeah,
Thank you.
okay. Thank you. Thank you.
Well, last call for questions, anyone? And I like that last comment by Luke Tayton. In any case, keep it up. I think it's fantastic that you came from another discipline, another field, and brought your training to bear and with this problem, because I think it takes some innovation to kind of look at some of these data from in a fresh way, and appreciate your,
Thank you.
help in developing HLA Guesser, and I would encourage people who listen later and on the call to check it out and see how they might use it as it continues to grow, and also their
Yeah,
data sets
sure.
continue to grow.
Yeah.
So thank you
Yeah.
again. Thanks again for
Thank
talking,
you.
Maria.
And
It was
see
a pleasure,
you.
and we'll
Thank
be in
you.
touch again. Have a great
Yeah,
rest of the
thank you,
day.
Monica.
All right,
Bye, you
bye-bye.
too. Bye.